Alrighty, good morning. We're gonna go into the actual crossing, the genetic crosses. So, um, what we're gonna do is we're gonna kind of review a couple of things first. The first set of vocab terms, which would be what an allele is. If you remember, those are the different versions of the traits, and we're gonna denote those with letters. So that'll be important here in a little while when we're doing the crosses. And then the two different versions of your alleles is going to be the dominant and the recessive forms of the traits or the alleles. If you have both, if you have one copy of the dominant allele, you will automatically show that dominant phenotype, or what you look like, because remember, the genotype is the set of alleles you have. So if you've got, maybe you got the dominant version from just mom or just dad, then you'll automatically show that, like the brown eyes that we kind of went over. And then if you have in order to show the recessive phenotype, you have to be carrying both recessive alleles in your genotype. So if you remember, like I said, genotype are the letters, and that codes for your phenotype, which is what you're going to end up looking like. So we'll go ahead and get started with a couple more terms. We've got homozygous dominant, which means that you have both dominant alleles. So if you, I didn't change that, I probably should have. So with that, you've got two capital letters. I don't know that I would use the letter S. Maybe use like the letter B or something, or T, something that you can definitely tell the difference between the capital and lowercase. So like we said earlier, the dominant allele is going to be capitalized. And that's just what we mean by homozygous dominant. Homozygous meaning the same, dominant meaning you have both copies of the dominant allele. If you are homozygous recessive, you are going to have both recessive alleles. So that means you got the recessive gene from mom and from dad. So in this case, it would be two lowercase letters. Again, I wouldn't use S. I'd use something that you can definitely tell the difference between capital and lowercase. So instead of the S's that I so poorly chose. Then if you're heterozygous means different. That prefix hetero means different or both. And so in this instance, you're going to have one copy of the dominant and one copy of the recessive. So you're always going to show the dominant phenotype, but you can pass on the recessive allele. So again, this is where I was talking about the other day. This is how two brown-eyed people could have a blue-eyed child. If they were both brown eyes and they were both heterozygous, meaning they both had at least one copy of that dominant brown-eyed gene, but they were both carriers of the blue-eyed gene, it wouldn't show up in their eye color, but they could both pass that on, and the kid could potentially have blue eyes. So, okay. The next one is the genotypic ratio. Sometimes you're going to see it called the genotypic frequency. That's kind of an older term, though. So usually anymore, you're going to see genotypic ratio, and all that means is we're talking about how many of the offspring are going to be homozygous dominant, to how many are going to be heterozygous, to how many of the offspring will be homozygous recessive. So 3 numbers. So it might be like a 2 to 2 to 0, a 1 to 2 to 1 ratio, or something like that. It will always equal 4, though. And we'll talk about this when we're doing some genetics problems here in a second, but you need to make sure that you include zeros as placeholders, because if you took it out, I wouldn't necessarily know which number you left out. So you don't ever have to label the numbers, because we automatically assume that you put them in the proper order. So we know that the very first number that we're going to come to in the genotypic ratio is going to be how many are homozygous dominant. So it's the same order every single time. Your phenotypic ratio or your phenotypic frequency is talking about how many of them are going to look like the dominant phenotype versus how many are going to look like the recessive phenotype. So it's, again, it's going to equal 4, because you're going to have, when we do our crosses, you're only going to have 4 options. So this is probably a lot of vocabulary that is very random and somewhat abstract, which is okay. When we start putting it in context with the genetics problems, I think it'll make a little more sense. So we'll go ahead and do 1. You don't have to copy this down, but in your diagram, on your set of diagrams, I've gone ahead and given you an empty Punnett square, and I've labeled it for you. The parents, or the I guess we'll call them the P generation in this case. The P generation is going to be up along the top and the sides of those 4 boxes. I mean, it's a square, obviously. It's where Punnett square comes from. And then, so your parents can go above and on the side. I don't particularly care if the male goes on the top or the side, or where the female is located. Generally, as I read the genetics problems, the first parent, whether it's male or female, I usually put on the very top. And then the second parent that I come to in the problem is going to be on the side. It's just a personal preference, but honestly, it doesn't matter to me which order you do it in. So, but what you're going to do, oh, and then in the middle there, you've got your offspring, or the F1 generation. Okay? There's 4 potential offspring. Obviously, you can, people can have, or plants can produce more than 4 offspring, but it gives you a % likelihood that your offspring will turn out with this particular trait or whatnot. So if I was to ask you to cross to make an F2 generation, you take those individuals from that F1 generation and put them now in a separate Punnett square, like I've shown you next to that, and you would make a, they would now be where the P generation was in the last square, and then their offspring would be the F2 generation. So when I ask you for those, you'll know how many crosses you've got to do. So if you go ahead and set up a monohybrid cross, oh, I don't know if I talked about this. I don't think I did. The reason why they're called monohybrid crosses, mono meaning 1, hybrid blend crossing of genes, so it's a 1 trait crossing, if you will. There are things, and we'll look at them briefly, not super in depth, but we'll look briefly at the dihybrid crosses. That's when you cross 2 Punnett square, or 2 genes, and that you end up with a 16 square Punnett square, so it's a little bit more complex, but we'll stick with the 4 squares. But so go ahead and in your notes or whatever, you can draw a blank Punnett square and use the alleles big G and little g, cross a homozygous dominant male with a heterozygous female. So I'll stop talking for just a second, but you probably want to hit pause, and then we'll go over what it is or how it should be set up. Okay, so you should have, like I said, I'm gonna put the first parent on the top. So you should have a Punnett square that looks like you've got two capital G's on top, 1 over each box. And then 1 along the side for the female. Heterozygous means that they're going to have 1 dominant allele and 1 recessive allele. So then what you're going to do is you have that set up. Now you're going to go ahead and do the actual crossing. So you're going to bring down the big G from the top. So you're going to put that 1 G in the upper left box, and then you're going to bring Dad's other big G down into the top right box. Then you're going to go to the left there where Mom's genes are, and she's got a capital G first, so you're going to put that in the top left box. and then you're going to put mom's gene over in the upper right-hand box too. So those two offspring would be homozygous dominant. Along the bottom, now we're dealing with mom's recessive allele. So just now we're gonna we'll go back and we'll start with dad. So again, drop dad's large G into the lower left box, and in the lower right box we're going to drop down that dominant G, but then mom's recessive allele is going to go in these boxes across. So they're both going to be, those two offspring would be heterozygous. So with these guys, you can basically say that they have these two individuals, for whatever gene this is, whatever G stands for, they have a 50% chance of having offspring that are going to be homozygous dominant, and they're going to have a 50% chance of having offspring that are heterozygous. They will not have any, or they should not have any children that are going to show that recessive phenotype. So, because there's no possible way for the kids to get that second recessive allele since dad doesn't carry it. So that's kind of how you set it up, okay? So now we're going to kind of add in some phenotypes with this. So again, you don't have to write down the problem. You can just do this off to the side in your notes somewhere. But we've got long tails on mice are dominant to short tails, or they're recessive. The short tails are recessive. By the way, I don't know if this is actually true. These are just genetics problems I made up. So what you're going to do is you're going to cross a homozygous dominant male with a heterozygous female. So go ahead and set up your Punnett square. And put the homozygous dominant male on top and the heterozygous female on the side. So when you do that, it's basically the same as the last time. I just realized that. Sorry. Normally when I do this, I catch it. So what you're gonna do now is I might ask you how many of the offspring are going to have long tails? So you would look at the kids because you've crossed them. None of them have both copies of the short tail gene. So every single offspring should have long tails instead of short tails. So hopefully that makes some sense here. So, okay, on the next one. Okay, now we're going to start doing the ratios. So go ahead, I won't necessarily read this one. So go ahead and set up your Punnett square and do the crossing. And I'll pause for a moment. So do that, and then we'll come back and do the genotypic ratio here in just a second. Okay. So you've got a genotypic ratio. You're crossing a homozygous recessive flower. So you're going to have two recessive alleles. I forgot to tell you which allele to use. So let's say you use B. So that particular one would have two lowercase alleles, or two recessive alleles. And then the one on the side, the other parent, that would be a homozygous dominant flower. So they're going to have both copies of the dominant allele. So two capital letters. And then you're going to cross them, and then the way it turns out is, hopefully, what you've gotten is that every single kid has one copy of the dominant allele and one copy of the recessive allele. So what our genotypic ratio is, remember that order is the number of homozygous dominant to the number of heterozygous to the number of homozygous recessive. So, so with this one, none of the offspring there in the boxes are going to be homozygous dominant. All 4 of the offspring are going to be heterozygous. And then none of the offspring will be homozygous recessive. So your ratio should be a 0 to 4 to 0. Like I said, you don't have to label it because we assume that you've put it in the correct order. So that's the genotypic ratio. When I ask you for both ratios, make sure you label them so that way I know that you know which is the genotypic ratio and which is the phenotypic ratio. So, okay. Let's do this one back here. Just kidding. So if I were to ask you on this one, what's the phenotypic ratio? I'm basically asking you, what are all the offspring going to look like? Are they going to look like, are they going to have purple flowers or are they going to have blue flowers? And since they all carry one copy of the dominant allele, they're all going to be purple. So your phenotypic ratio would be basically, in this case, I would be asking you how many of them are going to be purple, which are dominant, to how many are going to be blue. So in this case, your answer would be 4 to 0, meaning all 4 of the offspring would be purple, none of them would be blue. So that would be your phenotypic ratio if I were to ask you for that on this one. So, all right, try this one on your own. I'll go over it here in just a moment. So try this one. I'll stop talking, push pause, set up this Punnett square, and then hit unpause, and we'll go over the ratios. Okay, so you should have the heterozygous dad on the top, the homozygous recessive mom on the side. You end up with the top 2 being heterozygous and the bottom 2 offspring are homozygous recessive. So your phenotypic ratio is asking you essentially how many of the offspring are going to be long-winged versus how many will be club-winged, because long wings are dominant, club wings are recessive. So which way would you go? In this case, your phenotypic ratio would be a 2 to 2 ratio. 2 of them, the 2 offspring on the top, the heterozygous, would be dominant, would show that dominant phenotype of long wings since they both have the capital W. And then the 2 offspring on the bottom would be club-shaped, meaning they would be recessive. So it's a 2 to 2 ratio. If you want to do it for fun, I'll give you a second. Calculate the genotypic ratio really quick. So you should have gotten a 0 to 2 to 2 ratio. None of the offspring are homozygous dominant. 2 of them are heterozygous. 2 of them are homozygous recessive. So hopefully that's what you got. Okay. This is going to be the last little bit that we'll do today. We'll do gender. So this one's pretty easy. Sorry, still at school. This one's pretty easy in terms of it's going to pretty much turn out the same every single time. What the reason why we're doing this is later we'll be looking at sex-linked traits, which you'll need the genders. So with this one, you've got our genotype, or our two genes, you probably have heard of these. We've got different chromosomes. It's called sex chromosomes. For females, those are X and X. So we use that as the genotype. Those are the genes you literally inherit. And then your phenotype would be you're a girl. For a male, their genotype, their sex chromosomes are an X and a Y. They have one X and one Y chromosome. And so we use that for their genotype, and we say their phenotype would be male. So now here's the funny thing. You don't have, I don't know why I have this. So just go ahead and cross that really quick for me. So set it up in a Punnett square, put the mom on top and the dad on the side. Can't really do genotypic and phenotypic ratios. I'm not sure why I put that there, but so go ahead and cross those really fast. So in this case, you've got your top two individuals, your top two kids would be female, and the two bottom kids would be males. So this is why you have a 50-50 shot of having a boy or a girl. Okay? Okay, so the last thing we're going to look at today is a little bit on probability with gender. So you've got a man and a woman who want to have a kid. What's the probability that their first child will be a boy? So it should still be a 50% chance, because you just did that Punnett square there. And then what is the probability that their first child will be a girl? Again, you have a 50% chance. Now, there are two ways you can read this, and depending on how I word it, you can get two different answers. What is the chance that the second child will be a girl? So if I don't tell you what they've already had, technically they still have a 50% chance that their second child will be a girl, because you always have a 50-50 shot of having a girl or a boy. Now, let's say I told you that their first child was a girl. What's the chance that their second child will be a girl? Well, now you can take that one out, that top left girl out, and now you've got three remaining options here. So they have a 1/3 chance that their second child would be a girl. So if I don't tell you, they always have that 50-50 shot, but depending on what they have previously, sometimes that can alter their probability chances. So anyway, yeah. So okay. So this is going to kind of wrap up this first part. Tomorrow we're going to go into a few more different types of crosses. So hopefully this isn't too crazy. Again, this won't be on your quiz tomorrow, so don't freak out on this. And then when we're done with this, I'll give you some genetics problems to kind of practice and do on your own. So if you do have any questions, just let me know.